Hey there, Honors Biologist, Ms. Dreesen here. Today we are going to be taking notes on Learning Target 5, which in a nutshell is active transport. So the set of notes covers essential question 2, why and how do molecules pass through cell membranes? And Learning Target 5 focuses on active transport, including the sodium-potassium pump, endocytosis, and exocytosis, which are mechanisms for a cell to move away from equilibrium and transport molecules against their concentration gradient. Again, when you're doing your notes, I will ask you to write your own study questions, and the descriptive text is in black, so you don't need to copy everything word for word, and the critical text to add, I've bolded and highlighted in red. In terms of adding more content, sometimes you might want to add memory clues that I might say audibly or verbally, so keep that in mind as well. So let's start with a brief review. What have we learned about so far? We've learned about that cells use a form of passive transport to move molecules across the cell membrane. In passive transport, molecules move from a high concentration to a low concentration. So if you take a look at this diagram, here we can see there's a high concentration of this molecule in the extracellular or outside of the cell. So this is the extracellular fluid outside the cell, and you can see that over time, through random molecular movement, it gradually passes between these phospholipids, in this case, until it reaches about equilibrium, a balance of the number of molecules on each side. So this is going from a high concentration to a low concentration. This can also be referred to as moving down or along the concentration gradient. The word gradient refers to slope. So when you think about what this means, I want you to think of, say you have, for example, a slope, a hill. And at the top part of that hill, you have a lot more ground underneath, or a lot more space underneath in comparison to the lower end of the slope. So if you were to drop a ball at the top of this slope, what do you think would happen? Well, over time, that ball will naturally roll down the hill. You don't need to push it. And so this is what it means when it says that it rolls down or along the concentration gradient. So because of that, it does not require energy in order for these molecules to move from high to low. Just like if you put that ball at the top of the hill, it naturally rolls down without you having to push it. And the end goal here with passive transport is to achieve equilibrium, about an equal concentration of the amount of that substance on both sides of the cell membrane. Now, when we think about these descriptors, I want you to recognize that active transport is essentially the opposite of all of these. So active transport, in comparison, is when molecules move from low to high concentration. So if you take a look at this picture, I want you to draw a couple of balloons upside down and right side up, and then add a little channel here. And I want you to illustrate that we have maybe 1 to 3 molecules on this side of the membrane, and there's lots of molecules on the opposite side of the cell membrane, and we're basically pushing few to where there's already many. It's kind of like if you think about, say you're at a concert and you're standing at the back of the room and the area in front of the stage is super crowded. You have to push to get through to that front of the stage. Similar thing here, you're pushing few molecules to where there's already many. So this is considered to be going up or against the concentration gradient. So if we were to illustrate this in the context of our slope, remember that a gradient refers to slope, where we have lots of molecules at the greater end of that slope and few at the end. So if you put a ball here at the bottom of this slope, where there's low concentration, and you want to move that up, is it going to do that on its own? No, you have to push it. You have to use energy to move that ball up the concentration gradient. So in active transport, energy is required in order to move these molecules or push them to where there's already lots of them. And again, the goal here is to actually move away from equilibrium. And why would a cell want to move away from equilibrium? There's actually a number of really great examples that we'll talk about. So a few examples could be, say for example, you eat a donut and you have tons of sugar molecules in your stomach, but what if your sugar molecules in your stomach cells are already concentrated in sugar? Well, you still need to absorb those extra sugar molecules. Or maybe a cell just needs more glucose in order to fuel the cellular processes. So absorption of glucose is one example of where we might move sugar from where there's less sugar to where there's already a lot of it, because maybe that cell needs more energy. Another great example is when white blood cells might capture foreign materials, such as bacteria. your neurons rely on active transport. We're going to talk about the sodium-potassium pump and how neurons send electrical signals because of this process of exchanging molecules from where there's few of them to where there's already many in order to carry some kind of electrical current. Other examples is the release or absorption of enzymes, hormones, molecules, neurotransmitters, so on and so forth. In plants, it might be absorbing excess minerals and ions from soil, the transport of minerals and ions and water throughout the plant, so on and so forth. So passive transport is great for moving molecules in and out of a cell, but sometimes cells need to move away from equilibrium, and that's what active transport is for. There are 3 types of active transport that we are going to be learning about. 2 of them are very easy and straightforward, endocytosis and exocytosis. And the last one we'll talk about is a type of membrane pump called the sodium-potassium pump. Let's start with the easy ones. Endocytosis is called endocytosis because it's when things move into the cell, lots of things. This could be a large molecule, maybe it's a really big molecule that doesn't even fit through a protein channel. Or it could be just lots of things all at one time need to be absorbed into the cell. So endocytosis is when a large molecule or many of a molecule are absorbed into the cell at one time. So in your picture here, I want you to add arrows. So here we can see that the direction that these molecules are going is from outside of the cell to the inside cytoplasm. And what's really amazing about this cell membrane that we've learned about and how it's flexible and fluid is that it can basically just pinch inward and create new membranes. So here now we have a formed vesicle. We've learned about vesicles before. Remember that vesicles are like a cargo truck, or they carry packages around inside of the cell from the Golgi apparatus to the outside of the cell. But here we have the opposite. We are forming a vesicle from the cell membrane phospholipids, and then it's creating this. And now it's going to bring this to somewhere else inside the cell to be used. So in a nutshell, endocytosis into the cell. Some examples. So imagine this is a bunch of liquid. Here it says liquid. It could be hormones, proteins, and they're being absorbed into the cell. You can see it's pinching that cell membrane inward to carry it and form a vesicle. Here's what that looks like more zoomed in. So again, the cell membrane from the outside of the cell pinches inward. Oh, here's a really cool example. White blood cells. You have many types of white blood cells, which are the defenders of your immune system, and one of their jobs is to capture and eat, gobble up, invading bacteria or viruses, and they do this with the process called phagocytosis, which is P-H-A-G-O, and that basically just means they're big eaters, and so they gobble up invading things. Here's an actual microscopic video of this. This is one of your white blood cells. I think this is a macrophage. Macrophage literally translates to big eater, macrophage, and it's capturing and devouring a bacterial cell, and then it will digest it using lysosomes. Cool. Okay. Exocytosis is the opposite. It's the exiting or release of large molecules or large quantities of a substance out of the cell at one time. So maybe this is like maybe this is a hormone being produced by your pituitary gland, and maybe that pituitary gland cell is creating a ton of hormones and needs to push it all out at one time into the bloodstream. This is an example of that where, and I want you to add arrows here, so make sure you're adding arrows showing that we have a vesicle maybe coming from the Golgi apparatus, and then that vesicle forms with the cell membrane of the cell, and then it pushes those molecules out into the bloodstream. So again, I mentioned that this could be the release of neurotransmitters or hormones from glands throughout your body. And here's an example of a paramecium, which is a unicellular eukaryotic organism, and it's releasing, it's really hard to see. It's really this is the best quality I could find, but I thought it was pretty cool. So here's this little paramecium, and it's releasing, you can see it does a little fade. Yeah, so it's releasing molecules. Cool. All right, now let's talk about the more difficult one. So we've talked about endocytosis into the cell, exocytosis, exit out of the cell. But what about membrane pumps? Membrane pumps are a type of protein channel that requires energy to pump molecules against their gradient. So again, we're going from low to high, where there's few to many. And just like you would with a bike pump, you have to push, you have to put energy into that process of putting, maybe you're pumping extra air into your bike tire, you're putting air into where there's already a lot of it. And the molecule that provides this energy is called ATP. Now you've probably never heard of ATP before, and that's totally okay. We are going to learn more about this molecule called ATP next unit. For now, I just want you to think of ATP as like you know that glucose provides sugar. I want you to think of ATP as providing, or sorry, you've learned that glucose provides energy, but I want you to think of ATP as an energy molecule too. So think about it as like a $1 bill, where you can put a $1 bill into a machine and get something back out really quick. ATP is just like a $1 bill, an energy currency. So keep that in mind. So here's a really simple example of a proton pump. This is one example of a membrane pump where you can see here that we have a high concentration of hydrogen ions outside of the cell, and you can see there's a low concentration of hydrogen ions inside the cell. And so we're pumping hydrogen ions from where there's few to where there's many, and ATP is this energy molecule that's going to fuel this process to push that hydrogen from few to many. So this is a very simple example. I want you to add an arrow here, and I want you to add ATP here so that you can practice identifying the different parts. We're going to talk about a more complex example, which is the sodium-potassium pump. So sodium is represented by Na+, potassium is K+, so 3 sodium ions are pumped into the cell, and where there's already a high sodium concentration. So sodium is going from few sodium to high sodium, and then potassium ions are being pumped out of the cell from few potassium to high potassium. So they're going in opposite directions, but each of them is going from a low concentration to a high concentration. And ATP is being used to fuel or force this transfer of molecules. So I want to show, or why would this happen? Why would cells want to do this? Well, this process generates voltage. Because they have positive and negativity, it basically carries a signal, an electrochemical signal, along a cell. So if you think about the structure of your neurons, basically it carries that electrochemical signal from one cell to the next. And it's pretty amazing. It all has to do with the pump. So each of these is representing a pump that's pushing and transferring these in opposite directions to carry that. I want to show a quick video that illustrates this too. I want to show this one more time. So it creates an electrochemical signal that carries that. All right, so if you need to label this, please do. You've got 3 sodium going out of the cell, and we have ATP that's fueling this process. You can ignore this phosphate here, just ignore that for now. These 3 sodium go out and the potassium come in, and it's basically an exchange. Each of them is going from, I think there's a picture, I deleted the picture. Okay, let's watch this quick video.
The sodium-potassium pump is initially open on the intracellular side of the cell. It has binding room for sodium ions, 3 specifically. When these 3 sodium ions bind, the protein is phosphorylated. In our ATP video, we talk about what that means. ATP, a molecule of energy currency, transfers its phosphate to the protein. This actually changes the protein's configuration, the protein's shape. This shape change causes the pump to open to the outside. The sodium ions are pushed out to the outside, the extracellular side, and now, due to this configuration change, it's open to the extracellular side. It has 2 binding sites for potassium ions. 2 potassium ions bind. After the potassium ions bind, the phosphate group is released from the protein. The sodium-potassium pump reverts back to its original position, so it will open once again to the intracellular side. The potassium is dropped off inside the cell, and once again, there's room now for 3 sodium ions. The cycle repeats. We should mention that this pump is moving sodium and potassium against their concentration gradient. What do I mean by that? It keeps putting more sodium outside, where there is already a high concentration of sodium, and it keeps putting more potassium inside, where there is already a high concentration of potassium. So with this active transport, the ions are moving from low concentration to high concentration. A great example of active transport. So, what is the result of this pump's activity? Well, you are moving 3 positive ions out for every 2 positive ions in. So while this may contribute to the cell's more negative charge inside, it's really important to understand that this alone is not the big reason you see a voltage difference in the resting potential. The result of the pump's work means that there are specifically more potassium ions inside the cell and more sodium ions outside of the cell. This causes an electrochemical gradient. That means there's not just a difference in charge, but also in the actual ion types. Remember, in our diffusion video, we talk about how substances have a net movement from a high concentration to a low concentration. Well, if going by the chemical gradient here, those potassium ions inside the cell, they'd like to have a net movement out. They generally can't travel through the membrane directly themselves, as we mentioned, but they could if there happened to be a channel. These channels do exist. It is common for many cells to have far more potassium leakage channels than sodium leakage channels. This makes a cell membrane more permeable to potassium at rest.
All right, and that's more depth than we're going to go into. The thing I wanted you to focus on is illustrating, especially here. We have them moving from low concentration to high concentration. They just happen to be going in opposite directions across that cell membrane. 3 sodium out, 2 potassium in, both going from low to high concentration. All right. Now let's summarize. So I've given you a table in your notes. In this table, I want to see if you can do it from memory. So for example, what was the term that referred to when lots of molecules or very large molecules are being pushed out or are exiting at one time? What happens when we have a large molecule or many of something coming into the cell at one time? And here we even gave you the name in this picture. Briefly explain it. Just put a couple of words in here that briefly explains what's happening. You could even maybe explain why is it an example of active transport, and then give a couple of examples of use. And then second, in your review here, I want you to compare now passive transport, specifically diffusion and facilitated diffusion, to what we've learned about in this set of notes, which is active transport. So pause the video, fill these in, and once you've filled them in, you can unpause, and then you can see the answer keys coming up next. So go ahead and pause and work on them. All right, let's check your work, see how you did. So here's your answer key. So this first image represents exocytosis. This is when large or many molecules exit at one time. An example is the release of neurotransmitters or hormones. Here is an example of endocytosis. We have a large molecule or many molecules entering at one time. One example we talked about and saw a really great video clip of was a white blood cell capturing bacteria. And last but not least is the sodium-potassium pump, where sodium and potassium are pushed against their concentration gradient, and this process is fueled by ATP. And one example that was critical is nerve impulses in the nervous system. All right, let's review active versus passive transport. So active transport, yes, requires energy. Molecules move up or against a concentration gradient. It moves ions like sodium and potassium. It moves large molecules, and it can move small nonpolar and polar molecules if there are many at one time to move. Diffusion is pretty simple. It moves molecules down the gradient. Molecules can pass between the phospholipids, and because of that, it moves small nonpolar and polar molecules. Facilitated diffusion, this is the addition. It's, again, passive transport, so no energy required, but now it's being facilitated or assisted by a protein channel. So we're still moving molecules down the gradient. It always involves channel membrane spanning proteins. What that means is those transmembrane proteins that go all the way across that cell membrane. And because these are ions that have a charge, they can be moved using that protein channel in facilitated diffusion. And it can move larger molecules as well. So I hope this video was helpful. If you have any questions on anything related to active transport, feel free to come and ask me questions. Send me a message. I hope you're doing great. I'll see you soon. Bye.